Good morning. My name is Inaaya Shariq. I'm a junior advisory board member, and I'm a two-time osteosarcoma survivor and a hip-to-sarc amputee. Dr. Volchenboum is a professor of pediatrics and the Associate Chief of Research Informatics Officer of the Division of Biological Sciences at the University of Chicago. He is the Associate Dean of Master's Program, and he designed and launched the UChicago Masters in Biomedical Informatics. His clinical specialty is in pediatric hema and onco for caring of kids with cancer and blood diseases. His research group involves in UChicago's data for the common good, dedicated to building communities, platforms, and ecosystems that maximizes the potential of data to drive discovery and improve human health. Sorry about that. Okay, Chicago's Data for the Common Good flagship project and Pediatric Cancer Data Commons is dedicated to liberating and democratizing international data for pediatric malignancies. He's the Director of Informatics Core for the Clinical and Transnational Science Award, and he is the Director of UChicago's Clinical Informatics Fellowship Program. So please, let's give a warm welcome to Dr. Volchenboum. Thank you.
Thanks, Inaaya. Okay, I've got 7 minutes, and I'm going to try to be a little bit edgy here. So those of you who made it back in the room, listen up. So as we kick off this series of talks on data ecosystems, I want to take a moment before we dive into the science to acknowledge something absolutely fundamental: the role of philanthropy, and especially groups like MIB agents, in making this work possible. Let's be blunt. Research funding right now is a minefield. Federal support is uncertain, with budgets flat or even shrinking, and the political winds are blowing hard against science, sometimes targeting entire fields, or even the very idea of open collaborative research. Scientists are feeling the pressure, facing layoffs, and watching priorities shift away from what matters most for patients and progress. If we relied solely on government or industry, many of the bold collaborative cross-disciplinary projects we're discussing today simply wouldn't happen. And that's where philanthropy steps in. It's not just a gap filler, but it's a force multiplier. Groups like MIB agents aren't bound by short-term deliverables or political cycles. They can take risks, fund early-stage ideas, and support the kind of open data sharing and collaboration that accelerates discovery and benefits everyone, not just those who can pay for access. In 2023, I got the email every researcher wants to get from Christina, and no, I didn't think that you were a bot. It simply said, "Congratulations, your outsmarting osteosarcoma proposal will be funded." For my group, Data for the Common Good, which houses the Pediatric Cancer Data Commons, this award was pivotal for our desire to build and deploy an international bone tumors data commons. In June of 2023, I was presented with the award at the Factor Conference by Debbie and Pierre Grillo in honor of their daughter and osteoangel Annalay. We've since launched the Hibiscus International Bone Tumors Consortium and are working with groups all over the world to collect and harmonize and share data. Philanthropy has repeatedly shown that it can punch above its weight, catalyzing breakthroughs and supporting the next generation of scientists when other sources won't or can't. But let's not kid ourselves. It's not about generosity. It's about power. Who controls the data? Who decides what gets funded? Who shapes the future of research? In a world where data are currency and where access to data can mean the difference between life and death for patients, philanthropic organizations have the power and the responsibility to democratize access, to demand openness, and to insist that it serves the public good. So as you listen to today's talks, remember the work you're doing, the dollars you're raising, the community you're building, it's not just supporting research, it's defending the very idea that science should be open, collaborative, and in service of the common good. In this climate, that's not just important, it's revolutionary. Today's session is especially meaningful. We brought together a panel of international experts who are each pioneering collaborative data efforts in osteosarcoma research and clinical care. Our goal is to showcase a spectrum of approaches being used around the world and to explore how building networks, standardizing data, and sharing resources can help us all move forward. Now, you can read their full bios in the program, but briefly here are the speakers and what they will cover. Sandra Strauss is a professor of medical oncology at the University of College London. She's leading the UK National Cohort Study in Osteosarcoma, ICONIC. She's an executive committee of Foster and chairs the executive committee of the Euro-Ewing Consortium. And today, she's going to share key insights from this major national study reflecting on progress, collaborative achievements, and the future directions for improving patient outcomes. Katie Janeway is an associate professor of pediatrics and director of clinical genomics at Dana-Farber and Boston Children's. Her research is focused on precision oncology and bone sarcomas. She's the chair of the Children's Oncology Group Bone Tumor Committee. And today, she'll be sharing innovative approaches for directly engaging patients in rare cancer research, highlighting lessons learned and discussing the power of integrating community participation with genomic discovery. Emanuela Palmerini is a medical oncologist and professor of sarcoma research at the Miller School of Medicine and Sarcoma Sylvester Comprehensive Cancer Center in Miami. She's run more than 30 sarcoma trials. Since 2021, she's been the co-chair of the European Consortium for Osteosarcoma, Foster. Today, she'll share insights on how collaborative international networks such as Foster are making a difference in patient care and driving progress in osteosarcoma research. Lu Shi is a pediatric oncologist and deputy medical director of the Musculoskeletal Tumor Center at Peking University. She's interested in biomarker investigation in tumor microenvironment and osteosarcoma, and she'll share her insights on collaborative data panels for osteosarcoma, highlighting advances and opportunities for data-driven improvements in patient outcomes. David Jenkinson has 30 years of experience in oncology research. He joined LifeArc in 2023 to lead their efforts in childhood cancer. With careers in both academia and biotech, he's transformed scientific funding for several charities. And since joining LifeArc, he's established their strategy for the delivery of sustainable change in childhood cancers, established See Further and the Children's Cancer Therapeutic Consortium, and worked with the community to explore unmet needs in various cancers. And he's going to talk about outcomes of the therapeutic discovery workshops on osteosarcoma, highlighting community priorities, unmet needs, and new opportunities to drive drug development forward in this field. And finally, John Okello is the scientific portfolio manager at Breakthrough Cancer. With a PhD in molecular genetics and an MBA from MIT Sloan School of Management, he's in the unique position to understand how to organize and lead complex healthcare projects. John's passionate about contributing to Breakthrough Cancer's mission and its dedication to facilitating impactful advancements in the fight against cancer through radical research collaboration. I love it. Today, he'll present the ambitious Team Defiant initiative, which unites national research efforts to define actionable targets for osteosarcoma, revolutionary trial frameworks, and collaborative discovery. As you can see, each of our speakers brings a wealth of experience and passion for making real change through data sharing and collaboration. After their short presentations, we'll open the floor for discussion, so I encourage you to jot down any questions or insights you have and come up to the mic or enter them in Slido. So let's make the most of this opportunity to learn from one another, to share openly, and to advance our collective mission, harnessing data for the common good. Please join me in welcoming our first speaker, Sandra Strauss.
Right, I'd just like to start by thanking Christina and the organizers for inviting me to be here today. I've sort of wended my way and arrived a little later than I should, but I was really very honored to be able to be part of this wonderful meeting. It's my first FACTA meeting, but surely this is a unique endeavor, bringing the international community together really to make progress in such a devastating illness, but also with patients front and center of it. And I think that really shows the commitment to improving things for our patients. So I'm going to talk to you about our ICONIC project. Let me just, oh, sorry, I think I've done the wrong thing there. There we go. These are my declarations. So in the UK, we have a clinical studies group that is somewhat like the Children's Oncology Group, and for many years we've run clinical trials, but unfortunately, without any new agents coming through, we're becoming increasingly frustrated about lack of improvement for outcome for our patients, and also the fact that we are still giving such devastating and toxic chemotherapy with long-term kind of sequelae. So we developed this project really to try and accelerate the development of new therapies and look at new ways to improve outcome for our patients by simply doing two things. Really, first of all, bringing the multidisciplinary community together, all focused on osteosarcoma in the UK, to prioritize clinical and biological questions, and then to begin to answer them by setting up a prospective cohort to recruit our newly diagnosed patients of all ages across the country with clinical data and samples and inclusive of patient experience so that we can start asking and answering those questions. So this is the study design. We were very fortunate to get funding from the Bone Cancer Research Trust, one of the national charities in the UK. I think many of you are aware of the incredible work that they do and how much they support our community. It was funded in 2 stages. The first was a feasibility endpoint. Really, could we engage the community? Could we get everybody together to really recruit the patients to be able to answer the questions? We were very pleased to have met the criteria that we needed to do so, and this allowed us to leverage further funding through a very generous donation from the Hamilton family to continue this work, continue the recruitment, and then start funding some of the research objectives that we had. So the trial continued to recruit, and we just finished the recruitment in January of this year. And we recruited just under 350 patients. Because we have the National Health Service in the UK, we do have centralization of patient care with designated oncology centres through the country geographically, designated as pediatric or AYA or adult centres. We were able to get representative recruitment from each of those and our major surgical centres. So there are only 5 of them, and I think that's probably quite different to what happens in the US and many other countries. We were able to recruit patients representative of the population, including older patients and including some with some of those rarer primary sites like craniofacial osteosarcoma. We're beginning to get some insights in variation of clinical care across the country, across ages and primary sites, and also the use of systemic therapy. We in the UK have access to mifemertide, for example, for patients with resected osteosarcoma under the age of 30. The take-up and the use of that is about 2/3 of the patients have accepted to have that treatment. Perhaps that gives us an opportunity to understand a little bit more of their impact, albeit in a non-randomized manner. We also have a surgical team with a focus of a number of our studies looking at margins and response to chemotherapy. But with the use of an imaging repository, we now set up a team looking at some of those really difficult decisions about local management of osteosarcoma, particularly those patients who may require an amputation or some of the more difficult pelvic surgeries. And so this is a tumor board, a sort of virtual tumor board that's being set up and in collaboration with the Foster Consortium, so more widely across Europe, really to try and pinpoint some of the differences in our surgical decision-making and what impact that has on patient outcomes. We're also able to contribute to a local relapse study that Emanuela, who's sitting here, set up some time ago by having a patient cohort that has gone through the therapy. And then sort of moving on to the biology, so we've collected plasma samples on all these patients. We've got over 250 patients, and we've just kicked off a multi-omic analysis of cell-free DNA project led by my colleague Nishlin Pillay at UCL, really trying to look at some quite detailed nanopore sequencing, both looking at circulating tumor DNA, but some of the epigenetic events that occur in these patients. I think you heard some of that about earlier in the earlier talks. We've been able to contribute to some other pilot studies looking at circulating tumor cells, proteomics and genomics, and whether that may be another way to prognosticate for patients and have better insights in what may happen to them. And as I said a moment ago, we've also set up an imaging repository. So we've collected the MRIs and the staging scans on patients across the country. This has taken quite a lot of time to develop. I think many of those involved in these endeavors will understand this. And now for the first time, we're going to be able to leverage that. And we have collaborated with one of our scientific colleagues in UCL, really now to start looking at some of those radiomics that I know many people are doing, and perhaps try and institute some of the artificial intelligence to really try and understand more about the imaging and integrate it with the clinical data and the other aspects of the work that we're doing. Importantly, we have some biological objectives, two main ones, much like anybody else. There's been a challenge with having appropriate patient models for developing new drugs, and so we've harnessed the kind of group that has been put together to set up a pipeline to be able to get patient samples. We've got about 20 cell lines now growing from patients that can be used, and we've been developing explants that include the tumor microenvironment so that one can look at some of the kind of more immune-based therapies. And then on the other side, just really excited to be able to collaborate with Adrian Flanagan and Isidro Cortez Seriano, who I think many of you know. He's spoken here before, really trying to decode tumor immune crosstalk in osteosarcoma, looking at genetic evolution and immune evasion, which is clearly such a challenge for our group. And this is leveraging and adding to funding from the Osteosarcoma Institute and others. This work has just really started ongoing, but I think, again, our cohort allows us to extend and expand and validate some of those findings. And the second arm is harvesting tumor-infiltrating lymphocytes, looking to see if we can find mutations that may be immunogenic and, again, lead to new therapies. I know there's a session on kind of the immune-based therapies a little bit later. Finally, last but not least, we've really built patient experience throughout the work we did. We have patient advocates in our group that helped design it, and we've got two arms within this. One is looking at the diagnostic journey of patients. I'm not sure how it works in the US, but certainly in the UK, many of our patients have a lot of challenges getting the diagnosis and then getting to the right place once they do have the diagnosis. So we have a stream of work having a look at that, looking at time to diagnosis and what happens to patients, and then also looking at the quality of life of patients and the functional outcomes with all the challenging surgery and the chemotherapy that they have. So we've developed a new patient-reported outcome called SAM, which we are validating and comparing to some of the older methodologies, really to see if we can learn more about what makes patients have a better outcome and how we can improve that through the work that we do. And we've just got an analyst just started to do that. So really that's it. There's a very high-level summary, and I pose that this work is really only just starting. But I think we're very grateful to our funders to allow us essentially to learn from all the patients that we treat, and also foster collaboration. We can leverage funding to do further analyses, and really in line with what this talk is about is to support further research and provide this as a resource for the community. So I'm very keen to hear what everyone else has to say and what opportunities we have for sharing going forward. So I'll just finish by acknowledging many of the people that are involved in this, our funders and the Hamilton family, the patients and carers that have been involved, and all the many, many investigators and those who are actually doing the science. So thank you very much.
Great. Welcome up, Dr. Janeway.
Thanks so much. So I'm here to talk to you about directly engaging participants in rare cancer research and the Osteosarcoma Project. So first, this is my most important slide, which is to thank all of our participants and our advocacy partners. This project I'll tell you about really would not be possible without the very early engagement and involvement with many of the organizations shown here. In addition, I'd like to thank the members of our stakeholder advisory group and our Hispanic Latino focus group who really inform this research study. So the aims of the Osteosarcoma Project are to establish a network of engaged pediatric and adult participants with osteosarcoma to co-create a shared database of clinical, genomic, molecular, and patient-reported data to enable research. We collect medical records, patient-reported data really through surveys, archival tumor tissue samples, and saliva and blood. We aim to define the clinical genomic landscape of osteosarcoma, and here the sort of key concept is that although we understand quite a bit about the genomics of osteosarcoma, and you heard some beautiful work in the last session, Most of those data sets don't have deep clinical annotation, meaning they don't contain a lot of information about how the patients present, what treatment they get, and what the outcomes are. And so we have really not been able to define in osteosarcoma clear subgroups of patients that will respond well to our treatment or where our treatments will fail. And it's really this linkage of genomic data and clinical data through something like ICONIC and this project that will allow us to do that. And we're also aiming to share this de-identified data quite widely with the research community. And then finally, we're aiming to study and optimize the approach to direct participant engagement in cancer research. And we're doing this work through a network called or it's funded through a network, an NCI-funded network called the Participant Engagement Cancer Genomic Sequencing Network. There are actually 5 centers. And we are one center, and the Osteosarcoma Project is being run alongside another rare sarcoma project called the Leiomyosarcoma Project. So we're actually learning together about participant-engaged research. So I just want to, I've talked about this study before here at the Factor meeting, but I want to briefly review the study procedures. So our study is available online to anybody in the United States or Canada who has osteosarcoma at any time in their disease course. Unfortunately, because of sort of research regulation, we are not open to bereaved loved ones of osteosarcoma participants. However, you can register. And at some point, if we're able to build out a study like that, if you register, we'll be able to reach out. We have an assent for participants who are under 18. We conduct a baseline survey and then ask for medical record release and ask participants if they would like to receive back some of their genomic data. Once we have that information, the participant receives and returns a kit to collect germline, which is either saliva or blood, and then the research team reaches out to the institutions where the participants reports that they received treatment, and we ask for medical records and archival tumor samples. Once we have a germline and a tumor sample, we then proceed with the next steps. We offer genetic counseling and germline cancer predisposition testing with return of results to the participant, and we proceed on sequence tumor and normal. Very importantly, the genetic counseling and germline return of results requires that the participant make an appointment with Genome Medical, which provides the genetic counseling, and that notification is provided via email. The sequencing we perform is in collaboration with the Broad Institute. On the left-hand side, I'm showing you the CLIA, or return of results sequencing. The germline sequencing is a 70-gene panel test for common cancer genes. The somatic or tumor mutation assessment is done through paired tumor germline whole exome sequencing that's really interpreted like a gene panel. We have the sort of commonly affected genes in osteosarcoma selected for return of results. Then we also perform research sequencing. We do paired tumor germline whole genome sequencing. We perform tumor whole transcriptome sequencing, and then circulating tumor DNA. This all seems very neat and tidy. However, there's additional complexity. Sometimes we only receive a tumor, in which case we're doing a targeted NGS panel with our collaborators at the Brigham for return of results. Sometimes we only get a germline. If we only get a germline sample, we're performing whole genome sequencing. And because the 2 samples come from 2 different places, we have to wait 3 months to achieve pairing before proceeding with one of these unpaired approaches. And participants from whose care, meaning the sample originates in New York State or Canada, are not eligible for Broad clinical sequencing because they don't have the appropriate clinical testing certifications from those places. This is the somatic shared learnings approach. It's a limited list of genes that are reported, and we actually had a number of meetings with our patient community to figure out how to provide results of a genomic sequencing test directly back to patients without a doctor involved. And so we came up with some unique approaches and text around doing this. I'm showing you here the outreach that is maintained by the Count Me In team, which is incredibly robust. We do, I can't read that on the line, on the figure, but we do newsletter communications, we do in-person events, there's a ton of social media, And this is sort of really how we get the word out. And this is showing you from the start of the launch of the project in 2022 up until December of 2024. Here is our accrual from the launch of the project. Also, as I was saying, in 2022 in the fall. You can see a long, straight upline when we first started. That consists of 2 patient populations, both from our legacy Osteosarcoma project. So there was an initial project launched really with philanthropy, and that enrolled about 180 participants. 50 of those participants then consented for this second project that allow return of results. About 140 did not, but I'm counting both of those groups because we do have information from both of those groups. Since launch, we've accrued about 5 patients per month. We have 296 participants consented with releases signed. Please help us get to 300. The QR code for our project is there. Participant interviews that we've been doing to better understand this participant-engaged research tell us the biggest motivation is to advance science and help others, which is incredible. There are some quotes there about that. But we've also learned that it's hard to take this on because it's an additional burden that occurs when people are already experiencing illness and all of the medical care associated with that. These are the demographics of our enrolled participants to date. For sex, you can see that there's actually a majority female population, and then for the sort of race and ethnicity, it's primarily a Caucasian patient population, but we do have people of more than one race and also representing other ethnic and racial groups who get osteosarcoma. And the largest number of patients who are participating are between the ages of 10 and 19, but we have participants all the way up to age 60. Participants are enrolling from across the United States and Canada. This map shows all of the locations where participants enroll. And when we did a snapshot of the 153 participants for whom we had medical records, they had received care at 155 different institutions. This is a very complex slide that shows you of the consented participants who signed their medical record release, what types of samples had we received. It boils down to this. We received tumor samples in about 30% of patients and normal samples in about 50% of participants. And currently, we have 141 patients with germline samples and 136 tumor samples from 85 patients. So that's about 1.6 tumors per patient. Once you get those samples in hand, what the sequencing output is very complex related to what you have. One important point is that about 14% of archival tumor samples obtained from pathology departments across the country will not succeed to a sequence result. That's because sometimes they send us not enough tumor material. Sometimes that tumor material has been processed with harsh decalcification and won't proceed in the sequencing. Currently, on a sample level, we have successful whole exome sequencing of about 38 tumors, and we have another 78 in progress, and germline sequencing from over 100 participants. We did a genomic data freeze on December 31st, 2024, when we had 19 patients and 27 tumors sequenced. You can see that the male-female ratio is about equal. We have 79% of patients who had localized disease. Most patients had high grade, and about 40% of patients had progressed at some point. About half of samples that were sequenced were from recurrence. We are identifying the expected genomic variants from these archival tumor samples. And the bar graph are the genes that are most commonly returned to participants in the first 20 cases. You can see that we can identify complex genomics rearrangements, including in the whole genome's TP53 rearrangements. I'm going to skip this case, make just one more point, and then I'll stop. Just looking at the clinical data that we're receiving, we're doing both medical record abstraction and surveys. And this is showing a figure where each patient is a row, and each patient is on a row, and each drug received by these patients is in a column. And if it's green, we have a match or agreement between the medical record data and the survey data. If it's red, there's a mismatch. And not surprisingly, the mismatches are mostly that we don't have the information in the medical record, but the participant reported receiving that drug, which just shows you the importance of getting both types of data. We are ramping down the study. Our funding ends August of 2026, that should say, not 2025. We have end enrollment planned for December of 2025, which is coming up quite soon. We're messaging and planning a virtual enrollment event. And our current estimates are that we'll have about 330 participants enrolled, 175 tumors from 110 patients, 200 germline samples, and 85% of participants with clinical annotation. We're working on other components, and please come to our Research Bee, 8:30 to 10:30 a.m. tomorrow here at the MIB conference if you would like to sign up, or use the QR code. And with that, I want to thank this very large team that's required to conduct this research, and apologies, I did get flag shamed. Thank you so much.
Thank you, Katie. Next up is Dr. Palmerini.
Good morning. It's my pleasure to present the journey of the Foster Consortium. I'm a medical oncologist. I've been working at Istituto Ortopedico Rizzoli, and I'm now at University of Miami. And I'm presenting on behalf of all the group. Sandra is also active member, and especially Natalie Gaspar, which is the chair. So the idea to launch a European consortium came from an unmet need. Most of you will know that Ramos trial, European and American, got together all the pediatric and medical oncologists to get a randomized trial with more than 2,000 patients registered. Unfortunately, all the hypotheses weren't successful, and the network failed. So we wanted to build something new and something working. And the final goal is most important to all of us, to improve outcome in patients with osteosarcoma in terms of duration, but also quality of life. And I'm very proud to see this path improved over time. And I'm actually surprised because if I look back in April 2001, this network didn't exist at all. It was just like the first month when I received an email from Natalie Gaspar. Then all the signing documents were proceed. There was a virtual meeting, COVID hit, and then we had, in 2022, the first physical meeting in Paris. These are the numbers now. More than 330 members across 20 countries, and there is, of course, a higher proportion of Italian and French and Spanish and UK, but there are also representation of Greece and Lithuania, so I think that's very important. It cover-ups all Europe and the UK, plus UK, but also these disciplines are all there, from pedonc, but pathologists, bioinformatician, in order to get all the answers possible there. And you will see in the composition then of the working packages. We needed to give a structure, so we needed to build an executive committee, which has a representation from each of the country, but also a young investigator and a patient representative is in the executive committee, and also a biologist, a statistician, in order to get all in the decisional level. These are the working packages. The first two are biology, genomic, and preclinical, and then we have 3 clinical working packages, relapse, first-line trials, and local treatments, and then we have also radiomics, quality of life, and data sharing. After 1 year, we also added the working group number 9, which is working on genomic predisposition, and I really appreciate the work presented by Catherine Janeway right now, because I think it's really, really important, and we are also working on that. These are the leaders of all specific working packages, and we also elected secretaries among the youngsters in order to build for the consortium of tomorrow. And all this network is in place to collaborate. So the VUPs continuously meet online to get to collaborate and to bring ideas from preclinical to the proposal for the clinical trials. And these are the Young Investigator Committee. Again, we started to work a lot with them. I think they are very great resources, not only because they are a really hard working force, but because they can really represent this. In osteosarcoma, the trials last forever. I'm publishing now papers that were started in 2011. So collaborating with them give our ideas and project lifelong duration. And also, we needed a project manager, so we hired Esperanza, and I thank her for the continued support to all the group. And we needed also to have an identity, so we started to work on social media, but we needed a logo. And we had this original proposal to ask all the patient and patient representative to participate to a contest. And you can see in this picture the winner of this contest, and kind of sweet. I showed my son yesterday, and it's actually this picture, which was digitalized, but has been is our logo, and which represent Europe map and land looking at osteosarcoma. And also, you can find all the link for our referral. And we already built up on publication, on the recurrent osteosarcoma trial and prognostic factor review to see what is our starting point, but also collaborative paper. And Sandra is here. We did a couple of position paper, which is a tremendous effort in such a short time to get pediatric osteosarcoma recommendation in children and adolescents, but also, as you can see here, a position paper on bone tumor sampling. So bone tumors represent a unique challenge because you cannot use actually decalcified tissue, and also the bone is really, really strong to get tissue. So you need expert pathologists and the process that consent to preserve the genetic information, and this guide is really, really something that would last and would be available for all the future generation of pathologists that approach bone for the first time. It's very important. Quality of life, there are several groups working on quality of life, but the landscape in bone tumor is less well studied, and so we were able to collaborate with the quality of life working group, so to get this review, which will serve a starting point for the clinical trial, which I will share you now. So this is the master, we call this master file. The clinical trial, this at the center, is the randomized trial that we were able to almost start, and to finance. So the ATRAC grant will was in 2023 was awarded to the Foster Cabos clinical trial, and we will also collect tissue sample and radiomics to answer different questions. The idea is to include all patients without high-grade osteosarcoma older than 2 years, and after standard treatment, more than 500 patients will be randomized to placebo versus maintenance cabozantinib for 1 year. And I think that in a considerably short time, it's impressive that a trial was generated. So I really thank all the patients that were part of the generation of this consortium and the clinical trial path. And I thank also all my colleagues that constantly worked online and to this physical meeting. The one in the middle is in Bologna. And the next year will be in Warsaw. And these are all the founders and patient advocate. Without them, most of this project wouldn't be performed. And I thank you for your attention and for inviting me.
Next up is David Jenkinson from LifeArc.
Thank you. I'd just like to take the opportunity to thank MIB agents for the opportunity to talk to you today about the outcomes from our Pediatric Bespoke Therapeutic Discovery workshop in osteosarcoma. I want to take you through a little bit of an introduction about who LifeArc are, because I suspect there are a few in the audience that know that, how we've gone through some prioritization, the workshop itself, and the output from that. So firstly, LifeArc. We're a self-funded, not-for-profit organization in the UK, but with an international focus. We work in rare disease and global health, with particular interest there in infection. And about a year ago, we launched a challenge in childhood cancer. We work in the translational space, making new drugs and early testing of those in clinical trials. Back in January of 2024, we knew that we needed to do some prioritization around childhood cancer. And so with the cooperation of COG, ITCC, and PSYOP Europe, we did a survey of clinicians in the space. And we asked them a simple question. In your view, what is the biggest unmet need in childhood cancer? We allowed them to answer that 3 times. And what we didn't tell them was that if they voted for something in their own space, they got half a vote. But if they voted for something outside of their specialty, they got 1 vote. Off the back of that, as you can see, high-grade glioma was at the top. But there's a large number of unmet needs. Now, we know that we can't manage all of those, and even the liver tumors and low-grade gliomas at the bottom have unmet needs. But we need to be proactive in some and reactive in others. So we'll respond to information and to requests around the ones below the line, but we'll be proactive in the ones above the line, identifying the needs and reaching out to address them. So that brings us to the workshops. The purpose of these is manifold. It's to understand the epidemiology and the clinical standards that are currently in place. And this is part of the work that is being driven by the patient advocates within the Team Protect Cancer Grand Challenge. It's to discuss the scientific evidence and understand where we need to strengthen the biology. And we do that with partners such as CRUK and disease-specific partners, including in this case MIB agents. Thirdly, it's to understand the opportunities for novel drug discovery, and we do that through a collaboration that we've established with Cancer Research UK, Cancer Research Horizons, called See Further, which is there to really take targets and move them through to preclinical. And then finally, to understand what the current opportunities are in the clinical space, and we do that with ITCC as our partners. So there have been a number of workshops and a number planned for the future. As you can see, the osteosarcoma one was in February of 2025. And we run them about every 2 months. And we do that because it takes that long for the work behind them to be put into place. Once we've completed the workshops, We will publish. We're committed to publishing the outcomes. We're trying to find a place to do that at the moment, but those are being written up. We have identified and prioritized opportunities and proactively seek further information about that and those opportunities to make sure they move forwards. We can and do put CDAs in place to make sure that that's possible. And we work to inform our charity partners where there's work that is too early for us to fund and maybe where the science needs a little bit more strengthening. So the workshops themselves, for the osteosarcoma, we produced a target list of 125 targets, categorized by intracellular, cell surface, and some already in the clinic. The ones already in the clinic were moved into the clinical trials part of the program, but the others were prioritized by experts in the space, and including an additional 12 targets were brought forwards. At a pre-meeting, 6 targets were selected for further due diligence that were taken into the actual meeting. We use a scoring matrix which has been produced by ITCC. It effectively gives a RAG status, a red, amber, green status to a number of different areas. So, for example, with target presence, if it's been shown in multiple different ways in a large proportion of the patient samples, then it will get a green status. The targets that were prioritized to go forwards are shown here, and the clinical combinations that we discussed were around DNA damage repair and indirect targeting as MYC, as we've already heard, a very important mutation within osteosarcoma. So to the scoring, not expecting you to read this, but really it's just to show you that across the piece there was quite a lot of amber in this, and having done several of these now, we're probably seeing fewer greens than we might have seen in some of the other workshops, but certainly fewer reds, which is a positive. I think there's some definite opportunities here to strengthen some of the biology, but also to take some of these programs forwards. And that prioritization, which is then voted on by the experts in the workshop, identifies LRRC15 as the top priority, followed closely by SMARCA-AL1. And these programs are things that we are looking at. So to come to the outcomes and really talk about the conclusions that the workshop pulled up, in general, it was acknowledged that there are substantial unmet needs in osteosarcoma. We need to improve local control, increase survival whilst reducing toxicity, and make sure that clinical trials are accessible. There's a challenge because of variable backbone therapies, but a better understanding of targeting developmental pathways is probably needed as well, as many of the challenges that were identified were around targeting some of the proteins, but what would their toxicities be on normal growth? Around the targets, a conclusion was that LRRC15 is a good ADC target, although there was a discussion around the payload to be used on that, although recent publications suggest that MMAE may be an appropriate payload, which was questioned during the workshop. SMARCA-AL1 is a high priority target, and small molecule inhibitors and degraders should be explored for this target. ROR2 was another target that was identified for an ADC approach, whilst preclinical evaluation of KIF18A was felt to be important, but there are drugs available, and if that evaluation is positive, that should move rapidly into clinical trials. And similarly with the EIF4A1, a early phase clinical study would be warranted. RUNX2 was felt to be a potentially good target, but needed further preclinical validation before it was ready to move into drug discovery. Around the clinical conclusions, the direct inhibitor of MYC-OMO 103, the results from the clinical trials there should be awaited, but G-quadruplex stabilizers are also another approach. However, there needs to be a consensus on the levels of MYC identified for stratification. Around DNA damage response, biomarkers have not yet been identified. Biological studies are needed in early phase trials because what we're seeing is variable responses from this. And the results from the ongoing PARP and ATR inhibitor trials in osteosarcoma are awaited before pushing this forwards. But there is a potential for a DNA damage repair platform trial in this space. So this is done by a large group of people. Joe Baxter from LifeArc is probably the driving force behind it. He does all of the due diligence and is exceptionally thorough in that. Jan Molnar from ITCC leads on the scoring. The experts who helped us with this, the ones that helped with the selection and scoring in particular are in bold, but Natalie and Sandra helped with presentations around the setting the scene during the workshop. MIB agents as part of the workshop, but in particular I want to call out Walker Smallwood, who really set the scene from a patient perspective at the beginning of the workshop and helped us to take that forward. So thank you to everyone, and on to the next one. Thank you.
Thanks, David. And then finally, we have John Okello. So start getting your questions ready, because we'll take questions at the mics and through Slido as soon as Dr. Okello is done. Welcome.
Thank you, everyone. Good morning, everyone. I'm deeply honored to be here among such inspiring osteo warriors, dedicated families, brilliant researchers, physicians. I want to begin by thanking speakers before me and acknowledging the deep courage, insight, and partnerships that this community brings together. My name is John Okello. I serve as a scientific portfolio manager at Breakthrough Cancer, BTC. In terms of background, I'm a scientist with a PhD in molecular genetics by training, as well as an MBA from MIT Sloan School of Management, focused on healthcare innovation. I have research experience in cancer, as well as rare disease, where I've held various roles, both at the Hospital for Sick Children in Toronto, Canada, as well as Queen's Research Cancer Institute in Canada. As you can imagine, my background is both scientific as well as management. It's this background that attracted me to Breakthrough Cancer, which is an organization that is built around values that I deeply care about. That is radical collaboration, real-time data sharing, and a sense of urgency in tackling cancer. So today, I'm here to share with you how this model is being brought to osteosarcoma and how together we can accelerate hope. Breakthrough Cancer was founded to tackle cancers that have not seen meaningful breakthroughs in the past decades. And this includes glioblastoma, GBM, pancreas cancer, ovarian, acute leukemia, lung, and now we are focusing on osteosarcoma. Our belief is simple. We need to act urgently, think radically, and collaborate across all boundaries. And this may be institutional boundaries, disciplines, as well as geography. We think we can deliver better outcomes faster through this radical collaboration. Now, we still have these ugly statistics that every day more than 1,600 people die from cancer in the US alone. We all know that's far too many. How can we change the status quo? from breakthrough cancer perspectives, we think we can approach these barriers that are stopping, tackling cancer or curing cancer in three phases. The first phase is to build a team lab that would radically collaborate, leading to faster and more effective research outcome, and then scale these outcomes, things that are working and promising across diseases and institutions, with the hope of realizing the vision within a short period of time. And this vision is to build an ecosystem where translational science directly shapes care. For osteosarcoma, we are launching a team lab with an initial 4-year research plan. That's the first phase, proof of concept. We are not just trying to launch another team lab or research program. We are trying to prove a new way of doing science. And by this new way, our strategic goals are grounded in 3 pillars. The first is to intercept cancer by detecting and acting on it earlier. The second approach is by patient-driven learning, where we are using real-time data from clinical trials to guide discovery. And then, of course, at the center of it is radical collaboration, where we are building high-trust, high-speed teams across disciplines. These are not abstract ideas. These are already being implemented across our other team labs, and we think now it's time for osteosarcoma. And now you might wonder, what's this radical collaboration? What makes breakthrough cancer different? Now, in 2024, the president of Breakthrough Cancer, Dr. Tyler Jacks, as well as the chief science officer, Dr. Jesse Boehm, published an article on the 6 hallmarks of radical collaboration in cancer discovery, where, in a nutshell, the 6 hallmarks are showing that there's need for data sharing in real time, not just publications. There's also need for cross-institutional teams from both academia, biotech, and clinics to work together in a fast, iterative way, similar to what you see in high-speed performing startups. Of course, not forgetting that patients are at the center of all this. So this is our model of building team labs to tackle cancer. Now, the team lab is a virtual group of people who are at the core of the research engine that drives breakthrough cancer. Now, each is, as I mentioned earlier, is a high-trust cross-institutional collaboration. For now, we have funding for these team labs ranging from $10 to $15 million. Our funding institutions includes Dana-Farber, MSK, MD Anderson, Johns Hopkins, MIT, and more are coming in. BTC could not do it alone. We are actively welcoming experts from other institutions based on, of course, scientific interest, merit, and to fill in critical gaps. We have launched 9 team labs so far, and among it is a $25 million data science hub that helps in integrating data coming from the different disease teams. And the osteosarcoma team that is called Team Defiant will be our 10th and our first in a pediatric cancer. The vision of Team Defiant, I can summarize it in 2 or 3 ways. One is to ensure that every osteosarcoma patient receives biomarker-guided care, establish an integrated human and canine trial system, which will help to make osteosarcoma a more manageable disease. But the team, going over a period of brainstorming and based on their expertise, identified real barriers towards achieving this vision. And this includes things like samples. Biology is not very well understood. There are resources out there growing, but they're still largely siloed. And of course, the complexity of the osteosarcoma itself. And we recognize that there are trials going on, but some of these trials are very fragmented and limited. So BTC convened a series of multi-institutional strategy sessions starting from last year, to identify opportunities. And this is what led to Team Defiant being conceived, which is defining actionable targets for osteosarcoma. Team Defiant will focus on two tightly linked research engines. The first is an integrated trial framework, where a nimble adaptive platform will be launched combining human and canine co-trials with shared endpoints and molecular diagnostic to track disease. The second engine is target discovery, where a large data effort to aggregate and analyze over 600 osteo samples spanning human and canine will be done to identify vulnerabilities like surface proteins and so on, structural features like chromothripsis. And these engines will reinforce each other, accelerating both translation in both directions. Yeah, you might notice here, of course, these are the current team champions. More members are not in this deck, but they feature experts from our 5 funding partner institutions and beyond. Many of you know Katie, who is sitting next to me here, and other people in the audience. As you can see, there are those that are familiar to the osteosarcoma community, others are not. And this is because the others also bring in deep expertise that may not be available within the osteo community, with the BTC Data Science Hub at the center of this, making it uniquely positioned to foster radical collaboration. This is by design. We believe that the right team for osteosarcoma must have diverse and multidisciplinary and always evolve. So we are open to continue to build on this team as scientific needs arise and gaps are identified. Finally, I would like to, on behalf of Breakthrough Cancer, to announce that Team Defiant represents a $15 million commitment, and it's one of the largest osteosarcoma initiatives ever launched or to be launched. We are proud to build this in partnerships with other institutions, other stakeholders, foundations, and more to be announced. The formal launch will be later this year, but today is our first opportunity to publicly share this vision with this community. We and other stakeholders are here to listen, learn, and of course, move together with speed, humility, while we foster radical collaboration to make it better. Thank you for allowing me to be part of this community.
Okay, that was a wonderful set of talks. We have a couple questions already in Slido, but I'd like people to come up to the mics. I'm going to take that second question first from Brian. Thinking about AI approaches and the rapid development of new methods and techniques for analytics for large data sets, frankly, as a medical community, we're not prepared for these innovations, and we should be doing everything we can to get prepared to utilize these tools. So I'm curious if anybody can answer this. I certainly have my own thoughts. What should we be doing about the data that we're collecting so that we can apply these large-scale machine learning tools to our data?
I was very impressed with the iconic collection of radiology, because I think really what we should be doing is trying to collect data types that are where AI is most useful in analysis. So those in our world would be pathology slide images and radiology images. And I don't know, if Sandra, if you want to comment on how you're going about that. It's not the easiest thing to do.
No, thank you. I mean, I think it's a great question, isn't it? And AI, the word AI is used in many, many contexts, but essentially bringing all those data items together, that's the way that we are going to learn, but you need a huge amount of data to do that. And then it also comes down to the quality of the data and how it is kind of acquired. I think certainly what I'm reflecting on, having heard all of us speak, we're taking slightly different approaches, but really trying to get to the same endpoint. And then, I mean, we can talk to Sam, but how can we actually share our data between each other to get those numbers that we can do? And I think also looking at all the different studies that we're running, each of them has some strengths and others have weaknesses. And so it's only really by putting all that data you'll get enough to be able to work on it. I don't know enough about the sort of AI space, but I agree with Sam also that we need more scientists and people who are in that space to come and work with us to kind of leverage what we have acquired so far. So that's not really an answer to the question, but I think it's a very important one, and that we should do all that we can to kind of work together to get there.
No, but you both answered the question well, which was you need data and tools. And so harmonizing the data, getting good standards, developing sharing mechanisms, which is what you all talked about, is key. And then collaboration with our computer science partners and with industry that are doing this at scale and figuring out how to share data and how to make it available to these platforms are both going to be key. All right, we have a question back there.
Yes, good morning. My name is Ed Deezer. This question will be for anyone on the panel, but probably starting with Dr. Strauss. Great to see the collaboration of all the different specialties being brought together. Just curious if you speak to also, I didn't see an engagement on nutrition or neurology or behavioral health in that collaborative team.
Yeah, that's a really great question, and actually I think that sort of identifies how we set up the collaboration within the community that we work with. We haven't tackled many of those issues. I mean, I know that microbiome, for example, is yielding all sorts of incredible kind of breakthroughs in other cancers, and I'm afraid we haven't engaged enough with people outside our own sphere, and that, I would say, is a limitation. But maybe we still have those opportunities to do so, and kind of finding the experts who maybe want to work with us and learning from other cancers.
I could maybe answer with my children's oncology group hat on. So within the Children's Oncology Group, we actually have a very active behavioral sciences group, and also a group who are focused on patient-reported outcomes, which really gets at more of those sort of quality of life questions. We actually have tried incredibly hard to engage with them and have proposed as part of our trials measures of patient-reported outcomes and health-related quality of life. We have faced considerable uphill challenges in getting those adequately funded, in part because we lack data, which sounds counterintuitive, but when you're trying to do that type of research within the context of a clinical trial, you need a very robust hypothesis that you can either prove or disprove, and we unfortunately lack even the fundamental data around that. So I think it is very much a need in this field. I think that the treatments that we administer, either surgically or medically, are quite challenging and are inadequately studied and documented in the medical literature. I think it's a real need, and one that we really need to partner with the patient community to overcome.
I also want to add from the Foster Consortium, I think it's a very important specific data on nutrition. We do have a physiotherapist, and we will collect patient-reported outcomes and have a specific working group, but nothing specific on diet, although several pediatricians do monitor and use that consultation. At the Istituto Ortopedico Rizzoli, Dr. Longhi, one of our investigators, is collecting through questionnaire diet from bone tumors using an osteosarcoma, comparing with those of healthy people. So maybe those results will help us to learn on those differences, if there are any. Thank you.
I hope what everybody's appreciating is that standard clinical trials are not going to be the only answer to solving these problems, and that what you heard today from the speakers at getting at the deep real-world data that patients have access to through their electronic health records and through surveys and patient-reported outcomes and wearables and all sorts of other data are really going to be the key to trying to answer these important questions. I'm sorry, did you have something else, Dr. Strauss?
Yeah, I just wanted to add actually some of the challenges looking at patient experience. Certainly within our own cohort and across Foster, there isn't an accepted standard, as Katie said, and so it's quite difficult to build it into clinical trials. But I think there is an opportunity to reach a consensus. We've tried to do it in Europe to a certain extent, but we're still not there. But I think that is something that could be done more globally. Is to, if we're all asking the same questions, then we're gonna get closer to the answer than all of us doing things in a slightly different way.
Hi, Scott Sauer from Vujade Sciences. Great talks. I have a question that I think I'll originally direct to Dr. Okello, but could be for the entire panel. You mentioned kind of radical collaboration is at the core of BTC, and you specifically called out academic and industrial partnerships. How is BTC kind of working to facilitate those partnerships, and how do you envision those partnerships moving forward?
Thank you for asking that question. So, yes, BTC really champions radical collaboration, and this collaboration, like I mentioned earlier, is not only within academia, but also, includes collaborating with industry, like biopharma, as well as other stakeholders. So we, as I mentioned earlier, we have launched so far 9 team labs. The 10th will be the osteosarcoma team lab. All those team labs have industry involved, either by providing already approved drugs or drug candidates that are being used. So there's active clinical trials involving all the team labs. And some of those partners are providing in-kind contribution in terms of these are expensive drugs, and also logistically sometimes helping with shipping these drugs, which would be difficult for BTC or even our individual partner institutions to do. So it's at the core of of every team lab we build. But of course, how we approach this is there are 2 ways. First, the PIs or the team lab members themselves identify partners whom they think are good potential collaborators. Where there are gaps, we try to fill in and point them to certain companies or collaborators. This is the model that we build, but so I don't know if I provide that perspective well. Yeah.
So I just want to follow up on that topic and get other people's thoughts. So one of the things that is always hard to touch on is sustainability, and this idea of public-private partnerships and working with industry may be one of the keys to sustaining these efforts once the initial funding period is pushed through. I'm sure David and others have thoughts about this. So I'm curious how you all are thinking about the sustainability of your platforms once you run through your initial funding phase.
Can I go first? Yeah. So from Breakthrough Cancer, we are just like a 3-4 year old foundation. So our initial team labs are finishing their first phase, and almost all of them are going into the second phase, and this is where we are calling it the scaling phase. Of course, we recognize that we are just a small foundation. We might not be able to push from phase 1, phase 2 of clinical trials or development all the way to phase 3. So we are actively, working with other stakeholders to take on what is working from the initial funding. So this is our model. But of course, where there's need, we still come in stronger financially to make sure that we are not leaving teams that are making lots of progress, breaking through without, further funding. Yeah.
That's a really good question. I think the challenge in pediatric oncology as a whole is around the market failure that we see with the models for drug discovery and development. We're all aware of the opportunities that are there, in particularly with adult cancers, where there is venture capital and onward development by biotech and pharmaceutical companies. And we're obviously in this community very much aware of the success that Day One have had taking an individual drug forwards, repurposing that into childhood cancers. But I think the ability to actually build a sustainable model here is difficult. We need to find ways to make the sustainability not necessarily about financial sustainability. From a philanthropic view, the sustainability comes from impact. So we need to demonstrate impact, and if we can demonstrate impact in the things that we do, for LifeArc as a self-funding organization, it's less less important, but still important. But certainly for a donor-driven foundation, the ability to go out and say, you gave us money, we delivered a new drug for children, give us more money, is gold. So if we can do that, and if we can build other models that maybe talk to sustaining the infrastructure through returns, and what we should do, I'll caution, is not put financial barriers in the way of these drugs making it to children. So the more financial barriers, the more financial transactions we do, the bigger the barrier that is to actually, because someone ultimately needs to make these drugs, distribute them, and make them available. So from my perspective, that's key. How do we build a model that gets financial sustainability for the infrastructure, but relies on philanthropy and potentially low return money to actually bring these products through?
Thanks, David. We're going to squeeze in one more quick question, so it's going to be a good one.
No pressure. I'm Amanda Marinoff at UCSF, and it was really wonderful to hear all the collaborative efforts going on in so many different countries. And my question really is for all of you, whether you see opportunities to collaborate across country and continent lines, and how we might start to break down those barriers.
Yeah, thank you. I mean, that's such a great question. I think probably that's why we're all in this room, isn't it? To break down those barriers and work better together. I think now, having seen all the talks, I think probably we've got a responsibility to start working out how we can share. It's not so easy in the short term, but in the longer term, it's really worth that effort. I think what Sam's doing with the clinical data has been unbelievable. What we also want to do is build on that with all the genomic and other opportunities. I've been looking over to Lou. I mean, if we can get right across globally, I mean, that is a phenomenal opportunity for us.
As researchers and clinical investigators, we all want to collaborate our data together to do more things. Referring to AI and pharmaceutical companies and new drugs, we want to do that together so that we can use some new technology to spring out the targets, so then we can point to the targets to do more, drugs to this orphan disease. But sometimes the boundaries, sometimes politics things we need to overcome, and I hope later in our life we will have the opportunity to work together. Thank you.
I think that's a great way to end this. I think the boundaries are surmountable if we have people that are advocating and championing the efforts that we're doing. So we thank you to the speaker panel. Everything was wonderful, and thank you to all of your support. And let's go to lunch. Thank you.
Hey.